where we have stopped here. Base pair rule we have discussed. Always purine base pairs with the specific pyrimidine base, adenine with thymine, guanine and cytosine. How many hydrogen bonds? 2 hydrogen bonds between adenine and thymine, 3 hydrogen bonds between guanine and cytosine. And we have discussed, hence 2 chains or strands are said to be complementary to each other. These pairs are known as complementary base pairs. In this original double helical structure, one complete turn consists of 10 base pairs. What are the functions of DNA? What are the functions of DNA? First one is store and transmit genetic information from one generation to the next generation. How they store genetic information as a specific base pair sequence. Right? Now, as a specific nitrogenous base sequence, they can store the genetic information. Store genetic information for protein synthesis also. And protein synthesis. In the case of proteins, we also talk about nitrogenous bases. In the case of protein synthesis, we need the synthesis of proteins for our day-to-day life. We need proteins. For synthesizing proteins, the key player is this DNA molecule according to the nitrogenous base sequence of specific DNA. Actually, later we will discuss. If this is, if you can draw this one, in the case of nucleic acid, so genetic materials, actually, you may heard about these terms. Nucleus, chromosomes, DNA, genes, and these things you have, these terms you have heard. How we can make a relationship between these terms? Okay. Even a bacterial cell, even a plant cell, even an animal cell. To represent any kind of a living cell, I will draw a simple diagram for animal cell. If this is the whole cell, this is the nucleus. Actually, the cell is controlled by the nucleus. This is the nucleus. Nucleus, this particular organelle controls the all processes within the cell. Okay. Within the nucleus, we can see a dense mass. Actually, it is called as, you may heard about this term also, nucleolus. Within the nucleus, we can see the nucleolus. Within the nucleus, we can see the nucleolus. Other than the nucleolus, within the nucleus, actually, this particular membrane we called as nuclear membrane or nuclear envelope. As even we have represented this nuclear membrane or nuclear envelope as a single line, actually it has two membranes. Sorry, it is a two membranous structure. In the case of nucleus, under cellular basis, we will detailedly discuss about the nucleus. For now, you have to know every living cell has genetic materials, especially plants and animals. Every plant cell and every animal cell has a nucleus. This is a complex structure. It is an advanced structure also. Nucleus, presence of nucleus. in every living cells, we can see genetic materials. Genetic materials means DNA and RNA. In any kind of cell, we can see genetic material, but we can't see a nucleus in every cells. Presence of the nucleus means an advanced feature. Advanced feature, we can see such a nucleus, such an organized nucleus. Any question?
Notice.
Such an organized nucleus only in advanced animals. Advanced animals and actually advanced organisms like animals and plants. Other than if this is the cell, remember that this is the plasma membrane and this is the cell. Within the cell, we can see the nucleus. Nucleus contains nucleolus and genetic material. This is the genetic material. Genetic material. Normally, in advanced organisms, these genetic materials are appeared as chromosomes. Having chromosomes means it is an advanced feature. Having chromosomes. Yes, the structure of chromosome looks like this. You can see, you may have seen chromosomes are drawn, looks like this. Chromosomes. This is the chromosome. We can see DNA. We have discussed about the DNA. We know the DNA is a double strand, but we represent, for now, we represent this particular double helical structure as a single line. Within the DNA, within one chromosome, we can see only one DNA molecule, but it is extremely enlarged. Actually, it is very long structure, but it's still a single DNA. Remember that it is a single DNA. And within the chromosome, we can see DNA. Actually, these things we have to discuss in our molecular biology lesson. But I think it is better to tell you these basic things. They are not complex ones. Within the chromosome, we can see DNA. What is the difference between DNA and chromosome? DNA is included in the chromosome. Actually, in advanced organisms, later you will call them as eukaryotic organisms. Eukaryotic organisms. In eukaryotic organisms, we can see there are other types of proteins we can call as histone proteins. They are pearl-like structures. They are pearl-like structures. This particular DNA molecule is ringed around these histone proteins. This is the DNA and this is the histone protein. They are pearl-like structure, globular structures. They are proteins. They are histone proteins. What is the difference between chromosomes and DNA? Both DNA and histone proteins are included or compact in this chromosome. Yes. When we enlarge a single, actually each fragment, a small fragment of DNA, the structure looks like this. You know, this is a double helical structure. Double helical structure. There are some functional regions within each DNA. Actually, they are specific nitrogen-based sequences. Not all. Actually, we can't. These small regions we called as genes. Have you heard about genes? Genes?
Yes, teacher.
Yes. We can see genes, so many genes, so many specific regions in a single DNA molecule. Hari? There are some gene regions as well as non-gene regions in DNA molecule. Hari? That means these non-gene regions we can call it as introns. You will learn further in molecular biology. Hari? For now, you can understand genes are included in DNA. DNA are included in chromosomes. Chromosomes are included in the nucleus. Clear?
Yes, teacher.
Okay. Other than DNA, in chromosomes, we can see histone protein. Histone proteins. DNA plus histone protein. The structure looks like this. Okay, this is the chromosome. This is within each chromosome only a single DNA molecule. How it is, even how it is extended. Hari? Single DNA. DNA has some gene regions and non-gene regions. Actually, these genes are involved in our protein synthesis. Hari? Genes control each protein. Gene 1 may produce protein 1. Now you know genes are actually DNA fragments. Ne? Genes are actually some fragments of DNA to synthesize proteins to make proteins. Proteins are amino acid sequences.
Yes, teacher.
Yes, amino acid sequences. By the way, do you want to draw these things? Yes.
Should I draw them then?
Huh?
Should I draw them?
In unit 2, no need to understand all the things, but for your better identification, I suggest to draw these things. In molecular biology, we will discuss these things detailedly. Okay, you can draw this one. This is the chromosome. What is the relationship among these things? Genes, DNA, chromosomes, and nucleus. Genes are included in DNA. DNA are included in chromosomes. Chromosomes are included in the nucleus. There are some doubts among these terms. What are the genes? What are the DNA? Are there any relationships? Are there similar structures like that? Haven't?
No, teacher.
Yes, that is why those are the chromosome. This is a chromosome. Chromosomes within one chromosome, only one DNA is included. Not only DNA plus histone proteins. They make chromosomes. Chromosomes, 23 pairs of chromosomes are included in a single cell of human. 23 pairs means 23 into 2, 46 chromosomes are included. This is a chromosome. Or you can take a screenshot of this one. Later you can draw. Is it clear? Level up again.
A little.
Screenshot. Done?
Yes, teacher.
Yes. Other than that, how DNA molecules are involving in the protein synthesis. It is our problem. It is our matter. Yes. You know, in both DNA and in proteins, nitrogen bases are included. Actually, this is a complex story. I will teach only some simple points for now. It is a complex process. We have to explain it by using 2 or more days. Such a complex structure. Amino acid in proteins, we can see amino acid. There is a central carbon. You can if you can remember, in proteins, NH2 group, others.
Carboxyl group.
Hydrogen, R group, and COOH group. This is known as amino acid. And in the case of DNA, we are talking about nitrogenous bases. Nitrogenous bases. How we can do it? Pentose sugar group, nitrogenous bases, and phosphate group. Yes. How DNA molecules are involving in the protein synthesis is actually genes of DNA molecules are involving in the protein synthesis. And RNA molecules also have nitrogenous bases. Later you will learn in DNA, adenine makes hydrogen bonds with thymine, guanine makes hydrogen bonds with cytosine. But in RNA, we can't see thymine. We can replace thymine by uracil in the structure of RNA. Adenine with uracil, guanine with cytosine as previous. In the case of protein synthesis, what happened? These genes make commands for each proteins, each amino acids. In the case of protein synthesis, the process looks like this actually. In the case of protein synthesis, this double helix structure is unwound like this. And here we can see adenine, guanine, thymine, cytosine. Imagine this is a one gene region. This particular region has a specific sequence. What about the sequence? Adenine, guanine, thymine, cytosine. After separated this one, after separating these 2 strands, what happened? According to this sequence, RNA molecule is formed. How RNA molecule is formed? Uracil, then cytosine, then adenine, then guanine, and then guanine. This is called as, in the case of RNA, only a single strand. Yes, this is called as messenger RNA. This is called as, simply called as mRNA. Messenger means they will carry a message about the sequence of which type of amino acids should be bounded with each other. The initial command is produced by this particular gene. That is why we can say genes are involving in the protein synthesis. Each and every genes are specified to each amino acid. Then, according to this mRNA, there are another types. Actually, RNA are 2 types: mRNA, rRNA, and tRNA. According to the message of mRNA, tRNA molecules carry amino acids and Actually, this is happened in the ribosome. Protein synthesis is happened in the ribosome. Ribosome has two subunits. Actually, the amino acids are bound together by according to the command of mRNA, and this accumulation is done by tRNA. Right? It is the relationship between DNA, RNA, and proteins. I think you have a small idea about this process. Right? So now you can take an idea about this one. Store the genetic information for protein synthesis. Right? We are talking about function of DNA. Function of DNA. Actually, this is done by gene regions of DNA. Genes are involving in the protein synthesis. Right? Store and transmit genetic information from one generation to the next generation. First function, second one is store the genetic information for protein synthesis. And you can note this one also. Store and transmit genetic information from one generation to the next generation. Store the genetic information for protein synthesis. Okay.
Okay, teacher.
Then we have finished DNA. There is a small point to at this DNA part. Actually, these things we are discussing on molecular biology, but in your structured as a past papers, these things are combined together and ask some questions. And there is another point. Molecular biology. Yes. Write a small topic. Questions are looks like this. Here in the molecular biology part, here also they have discussed the structure of DNA molecule. Therefore, please take a screenshot of this and you can add these things to your notes. That means missed things. Are there any missed things in our unit 2? You can add. Therefore, take a screenshot of this part. DNA
Okay,
double
teacher.
helix model. Okay.
Yes, teacher.
And there is another one. There is another topic. Most questions depends on this. Write your topic as reasons of DNA. Reasons of DNA. Reasons of DNA to act as the vital genetic material. Vital genetic material. vital genetic material. First reason is accurate replication of DNA. Accurate replication of DNA. Accurate replication of DNA. Second thing is transmission of genetic material. Second one is transmission of genetic material. Transmission of genetic material from one generation to the other. From one generation to the other. What we have written, accurate replication of DNA and transmission of genetic material, genetic information from one generation to the other. Transmission of genetic information from one generation to the other. Actually, this is the genetic material. Transmission of genetic information from one generation to the other and ability to store genetic information. Ability to store genetic information. Ability to store genetic information. Last one is express hereditary information. Express hereditary information. Yes, that's all about DNA. Now we have to move on to the RNA. You need a small break or no?
Yes, sir.
Yes. Now our topic is RNA. Even DNA molecule is appeared as a double strand. RNA molecule, even it is folded or any, like, if you can remember, in the case of tertiary structure of proteins as well as the secondary structure of proteins, even it is how much folded, and still it is a polypeptide, single polypeptide. Like that, even how much folded, as they have some foldings in the structure, and even there are some intramolecular interactions between these strands, starting from here and ends with here. That means still a single strand. And even it is folded, or even it is even there are some interactions are formed, still it is a single strand. Remember that RNA molecules usually appears as a single strands. Clear? First thing is first difference of DNA and RNA. What is the first difference? In DNA and RNA, first difference is here deoxyribonucleotide, here ribonucleotide. Other difference is here thymine. Thymine is presented, here thymine is absent. Here uracil is absent, uracil is present. We can match our answer according to the type of questions. As they have asked about differences, we have to enlarge our answer like that. Anyway, can you hear me?
Yes, teacher.
Anyway, RNA molecule looks like this. It is a single strand. ribose sugar, adenine, guanine, uracil, and cytosine. There is a phosphate group. Phosphodiester bond looks like this. No hydrogen bonds between two strands. They can make hydrogen bonds with now is it okay?
No, teacher.
Still this sound is hearing the
Now okay, teacher.
RNA molecule looks like this. It is a single strand. No double strands. It is a single strand. The fourth difference is RNA molecule is appeared as a single strand. DNA molecule appears as a double strand. The whole structure. No need to. Yes, you can draw this one. RNA molecule looks like this. You know, actually. Here, adenine, guanine, uracil, and cytosine. It should not thymine. The other one is there are three types of RNA. Three types of RNA: mRNA, tRNA, and rRNA. rRNA. We will consider the note. Here you can see structure of RNA. This is a normally single-stranded nucleic acid composed of ribonucleotides containing which type of bases? Uracil, cytosine, guanine, and adenine. Complementary base pairing between two RNA molecules or within the same molecule may occur in some. That means even it is a single strand, after folding a single strand, there may hydrogen bonds. Then we have to follow the base pair rule between nitrogenous bases which are contained in a single strand. Therefore, they are intramolecular interactions. Can you understand?
Yes, teacher.
Previous case, we have discussed hydrogen bonds between two strands, two separate strands. Therefore, such bonds are intermolecular interactions. Hydrogen bonds are intermolecular interactions. The DNA strand is a molecule, type of molecule. Therefore, previous case, intermolecular interactions. In the case of RNA, always we can see only intramolecular interactions. Yes. Complementary base pairing facilitates three-dimensional shape essential for their functioning. According to their function, there are three types of DNA, but we can't see types of DNA. But in the case of RNA, three types of RNA can be identified: messenger RNA, transparRNA, transferring RNA, ribosomal RNA. And here you can see the structure of tRNA. And here it is a 3D structure. Even it has a 3D structure, even it has folded in some regions. Why it is actually the amino acid is carried by this RNA in the case of protein synthesis. There are some positions to attach to the amino acid. That is why such a shape is formed by tRNA. Depend on their function, their shapes are differ to each other. Okay?
Okay, teacher.
Even it has a 3D structure, even it is folded in some regions, still remember that it is started from here 3N and this is the 5N. As a DNA, they have also both 3N and 5N. This is the 3N and this is the 5N of a single RNA molecule. Even it is folded, even it has made some interactions, they are hydrogen bonds actually. They are actually intramolecular interactions. Clear?
Yes, teacher.
Yes. Then three types of RNA molecules can be identified. First one is messenger RNA, as I have previously mentioned. In the case of protein synthesis, according to the command by gene, the command is carried by messenger RNA to the location of protein synthesis. That is why they are called as messenger RNA. Messenger RNA. Messenger RNA is a linear molecule like that. Linear molecule. It is the least abundant type of RNA in a cells comparatively. අඩුවෙන්ම තියෙන එක තමයි. يعني abundance එක ගත්තොත්. There are two functions of messenger RNA. Copies the genetic information stored in DNA molecule as a sequence of nitrogenous bases. Second one is transports genetic information from nucleoplasm to the site of protein synthesis. That means, you know, DNA is included in the nucleus within chromosomes. Chromosomes. So then DNA are included in the nucleus. Actually, protein synthesis is happened in ribosomes, right? Related to the ribosomes. You know, ribosomes are embedded in the cytoplasm of the cell. That means exterior to the nucleus. The command is included in genes in DNA. Right? The command is carried from the nucleus to the outside. Right? That means the place of protein synthesis. This messenger carrying is done by messenger RNA. Copies the genetic information stored in DNA molecule as a sequence of nitrogenous bases. Second one, transport genetic information from nucleoplasm, means this matrix within the nucleus. We can see a matrix. This matrix we called as nucleoplasm, like cytoplasm. Cytoplasm in the cell. The matrix of the nucleus we called as nucleoplasm to site of protein synthesis through nucleopores. Nucleopores means this membrane, it is a porous one. There are some openings to go this mRNA. Second one is transfer DNA, smallest RNA molecule, linear, but it is folded and form three loops. First loop, second loop, and the third loop. Here there is a specific site we will learn further. This is specific site we called as anticodon. Anticodon. Our smallest RNA molecule, linear, but forms three looped structure as shown in this diagram. You can take a screenshot of this tRNA molecule also.
Okay, teacher.
Then ribosomal RNA, third type of RNA, they make ribosomes. Ribosomes are made by RNA. The ribosome forming RNA we called as ribosomal RNA. Ribosome is the location of protein synthesis.
Teacher. That means these ribosomal RNA are the ribosomes in the cytoplasm.
Ribosomal RNA, the collection of ribosomal RNA is, we can consider it as a ribosome. Actually, this ribosomal RNA create a ribosome. It is the most abundant type of, very good question. Hari ribosome structure, there are two separate ones, large. Actually, they are called as subunits. Each subunit is made up of RNA. This is the large subunit and small subunit. Hari, between these two subunits, polypeptide is formed. That means a protein is formed in the case of protein synthesis. And therefore, all the three types of RNA are involved in the protein synthesis. It is the most abundant type of RNA. Which one is the less abundant RNA?
Messenger RNA.
Very good. Very good. Messenger RNA is least abundant. Most abundant type is rRNA. How we can write? Simple R, capital RNA. rRNA. RNA. And RNA has a complex irregular structure. It provides the site where polypeptide chains are assembled. That means polypeptide chains means proteins. Proteins are synthesized. We can end this part. Therefore, please focus on this too. Okay. The difference between DNA and RNA. DNA is a double-stranded molecule, while RNA is a single-stranded molecule. DNA consists of adenine, thymine, guanine, and cytosine, absence of uracil, while RNA consists of adenine, uracil, guanine, and cytosine, and absence of thymine. Sugar molecule in RNA is ribose. DNA it is deoxyribose. Those are the things regarding nucleic acids. Where we have stopped here, RNA. Yes, you can complete your note. Structure of RNA. Initially, draw this diagram. This is the structure of RNA. It is a single strand. The difference is adenine, guanine, uracil, cytosine.